use of PSC101 for cloning. Although the cloning with PSC101, it is not so advantageous as we have discussed, because the selection of transformants, it is not easy on the basis of tetracycline resistance. But still, this plasmid PSC101 has been used to clone some genes from the plasmid of other organism like P1258, and this plasmid, it is present in gram-positive cocci like Staphylococcus. So genes from Staphylococcus plasmid, it can be cloned in PSC101 and then can be expressed in E. coli. Plasmid P1258, it contains several determinants or traits that can be expressed in the recipient cells. These traits includes like ampicillin resistance, erythromycin resistance, and resistance to certain heavy metals. इसमें मरकरी आ जाती है और कैडमियम आ जाती है. तो ये सारे ट्रेट्स जो हैं ये स्टेफाइलोकोकस के प्लाज्मिड पे होते हैं. और जब हम स्टेफाइलोकोकस प्लाज्मिड को, जो कि P1258 है, इसको डाइजेस्ट करते हैं EQR1 से, तो इसके 4 फ्रैगमेंट्स बन जाते हैं. That can be separated on agarose gel electrophoresis. So the plasmid DNA from PSC101, it can be digested with EQR1 restriction enzyme, and similarly the plasmid of Staphylococcus, it can also be fragmented by using EQR1. When they are ligated together, then the transformants can be screened for the presence of antibiotic resistance genes, that is tetracycline from PSC101 and ampicillin from P1258. How it is possible? These diagrams, it showed the cloning of Staphylococcus aureus genes that contained on plasmid 1258, P1258, and it contained different type of selectable markers like ampicillin, erythromycin, gene for mercury resistance, and cadmium. Whereas on PSC101, the resistance, it is for tetracycline. Here it is T. The resistance, it is for tetracycline. So both they can be digested with EQR1. So in this case of P1258, 4 fragments are produced and only 1 linear fragment for PSC101. Then they can be ligated with the help of DNA ligase. Here they are ligated, and then they are screened for the presence of antibiotics. Like after introducing ligated DNA into E. coli, then they can be screened for the presence of ampicillin, ampicillin, and ampicillin. So in some cases, there will be no ampicillin resistance, and where ampicillin resistance present here, the transformants, they are further screened for tetracycline, so that the genes from Staphylococcus aureus plasmid, for example, ampicillin resistance, it can be combined with tetracycline resistance from PSC101. So this is one of the example of the cloning of genes from other plasmids into PSC101.